We've talked about the BRCA mutations, which are increasingly understood that, getting testing for BRCA1 and BRCA2 is relevant, highly relevant. But then there's this conversation about HRD and then HRD testing. Mike, you want to sort of talk about that a little bit?
Yeah, so this is an important topic, particularly after the ESMO clinical trial presentations. Full disclosure, I'm still a sequencing kind of guy. I use panels. I want to look for Fanconi genes and their mutations. But you should be aware that there are at least 2 HRD assays, one by Foundation Medicine, one by Myriad, and they're based upon looking for loss of heterozygosity within the tumor. And the basis of that is that presumably if you have a tumor that has homologous recombination problems, you're going to lose lots of pieces of DNA. And so you see high LOH scores. And the Myriad assay adds something called telomere imbalance to it, and also large-scale transitions, which are just other measures of damaged DNA. These scores, a lot of individuals, I don't use them that much, but my colleagues do, and they certainly correlate with an increased benefit from PARP inhibitors. And I think the ESMO presentations really reinforced this. So we're going to be seeing individuals physicians using these assays more and more. The final point I would make is because you're looking at what's called a genomic scar, the loss of heterozygosity, that's permanent. So if you've got a patient on a PARP inhibitor, they grow through the PARP inhibitor, they're now resistant. The HRD assay is of no use because the scar remains. It doesn't really predict in recurrent disease after exposure to PARP sensitivity.
Well, two things. One, I think it's important to understand that you cannot always rely on the sequencing to look for germline tumors. You will miss some germline tumors if you rely on Foundation or Caris. So they do need germline testing based on a high-quality panel. In addition to somatic, right? So the germline's probably 10 to 15%. The somatic is about half of that. SOLO-1 included somatic mutations, but there were only a couple in the entire trial. So we don't have high-quality data on that, but we don't know of any data that suggests they don't respond just as well to PARP inhibition. So, I guess these tests are how you sequence them and how you pay for them is the other issue.
The only point I would add to that is, and we're going to talk a little bit about the recurrent setting later, but as of today, FDA approval does not require this testing. It's approved, the PARPs that are approved broadly in the recurrent setting that may or may not continue in the frontline setting. So it may actually be this type of testing may be required as per the FDA. That's not been decided yet, but certainly some of the data suggest potential relevance. So I think we all need to sort of stay in tune.
Well, actually, though, we was it this week that we got a indication for HRD positive patients who were
in the recurrent setting.
In the recurrent setting, based on the Quadra data. So again, this changes so fast; it's hard to keep up with.
Yeah, and again, this is a role for the pharmacist here, who's this is just so complicated right now, and an individual doctor wants to use a particular drug and to say, these are the FDA indications, and another drug may have a different indication, even though you're sort of in the same clinical setting, and that this all becomes very important. Unfortunately, it doesn't become less complicated.